Hi everyone. So I just wanted to take a moment to review mitosis with you, the steps of mitosis. Just looking through some of the quiz scores, like the online quizzes I've given you, the game. It looks like there's still some confusion. And even if you don't think you're confused, there might be some questions or some things that you misunderstood. So I went over all of this in the screencast from last week, but I just want to go through it again. I'm going to draw everything out, go step by step. So even if you think you get it, please watch this. There might be something you thought you understood, but actually misinterpreted it. So I'm going to go over everything again one more time, okay? I'm going to start with G1 of interphase. This cell has a nucleus, a nucleolus, one centrosome with two centrioles, and it also has all the other organelles as well. Remember, a cell has the mitochondria, the ribosomes, the Golgi apparatus, all of that is also in there. I'm just only showing the ones that are relevant to mitosis. And then the star of the show, there are chromosomes. I'm going to draw two chromosomes, one red, one blue. These chromosomes at this stage are not condensed and they are not duplicated yet. All that happens during G1 is the cell grows. So I'm going to show the cell growth, okay? So this will now be S phase of interphase. So during S phase, you still have the nucleus, you still have the nucleolus, and you still have that one centrosome. What happens here is each chromosome gets duplicated. So it gets its sister chromatid. So all that stuff we learned about a couple weeks ago with helicase, DNA polymerase, semi-conservative replication, all that goes on here. That's S phase of interphase. So you start with one copy of each chromosome, one copy of red, one copy of blue. And now you have two copies of red, two copies of blue. But the tricky thing here is that this cell has two chromosomes. This cell still has only two chromosomes. They're just duplicated. So I know that's really confusing, but we say that this cell has two chromosomes. This cell has two chromosomes, each with two sister chromatids. So the result of DNA replication is, yes, it's technically another copy of the chromosome, but we just call them sister chromatids now. So two chromosomes, one red, one blue. Then we go into G2. G2, the nucleus stays the same. You have your red duplicated chromosome and your blue duplicated chromosome. What happens here is a lot of the other organelles reproduce or get copied. Proteins get copied. A lot of creation of protein happens here. And really one of the more important things that happens here is the centrosome gets copied. Okay, so you now have two centrosomes, each with two centrioles. So this is G2 of interphase. Remember, there's a checkpoint here. So this is all of interphase. And there's a checkpoint right here that says, is this cell ready to divide? Are there any mutations in the DNA that occurred during DNA replication? Do we have enough proteins? Are the proteins we need present? Are all the organelles there? Are there two centrosomes? Things like that. So there's the checkpoint right here. And if it passes the checkpoint, that cell will go into M phase, the next phase of the cell cycle. M phase starts with prophase. We break prophase into two parts. Early prophase and late prophase, just because so much happens here. And you have to remember, everything's moving, right? This is a fluid cell carrying out functions and, all that good stuff. So things are moving here. So things start in early prophase that then finish in late prophase. So both of these are prophase. Okay? So in early prophase, that nucleus starts to break down. The nucleolus goes away. Nothing happens to the cell membrane here. It's the nuclear membrane. The nucleolus is gone, and those chromosomes condense into that classic X shape you're used to. A lot of things happen here. The centrosomes start growing those spindle fibers, and the centrosomes start migrating towards opposite poles of the cell. So these centrosomes start moving towards opposite sides. So a lot happens during early prophase. And all of that is finished up in late prophase. So by late prophase, the nucleus is gone. The chromosomes are all the way condensed. The centrosomes have reached opposite poles of the cell. So there's one on this side, one on this side. And the spindle fibers attach to the centromere region at the kinetochore of each chromosome. So there, the centromere is this, the part where the two sister chromatids are attached. They are attached by the kinetochore, which is kind of like a protein complex. And that kinetochore is also what the spindle fibers attach to. Okay? So a lot happens there, right? This really gets everything going. All right? Then the cell is going to transition into the next phase, which is metaphase. By metaphase, you have your centrosomes still on opposite sides of the cell, and your chromosomes have lined up at the center of the cell, right? They are at the middle. Metaphase is at the middle. The spindle fibers have pulled them there. The spindle fibers are what do the moving. So they attach to the chromosomes here, right, and start yanking them. So the chromosomes get pulled to the middle of the cell by the spindle fibers. So this is metaphase, characterized by the point at which those chromosomes meet the middle. Next, the cell goes on to anaphase, in which you have your centrosomes, your spindle fibers, and separated chromosomes. So this chromosome has been yanked apart. This chromosome has been yanked apart right there. So this spindle fiber is attached to the kinetochore. This one's attached to the kinetochore. They pull and they yank and they separate the sister chromatids that were once joined at the kinetochore. Same thing with this one. It yanks right there, right in the middle, pulls them apart and separates them. Okay, this is one part where you really need to watch an animation. I included an animation in the hyperdoc from the day you learned about the phases of mitosis. If you're a little stuck, I recommend going back and watching those animations. You really need to see this happen visually, right? Something happens between this and this. These chromosomes, these spindle fibers, everything's moving, okay? So it can be a little bit confusing if you're not seeing it happen visually, all right? Next, we get to telophase. In which the chromosomes have reached each side of the cell. They've reached opposite poles. The nuclear membrane starts to reform. The nucleolus starts to reform. the centrosomes are still there, but each side now only has one, right? This side gets this one, this side gets this one, and the spindle fibers go away. So telophase basically undoes everything that happened in prophase. And then you end with cytokinesis. So cytokinesis is when you get two separate cells, right? This splits. In animal cells, you get that actin ring that forms here and starts to pinch, and you get the cleavage furrow. And then in plant cells, the cell plate is built there, which is basically just a cell wall separating the two sides of the plant cell. But in either case, you get two separate cells. Each cell has one centrosome. The nucleus is back. The nucleolus is back. And the chromosomes start to uncondense. I'm actually going to show that happening back here. I'm going to show the chromosomes start to get back to that chromatin form. They're kind of like in the transition period here. They start to uncondense. By cytokinesis, they are totally uncondensed. They are back to that unduplicated chromatin form. And if we look at what we started with, we have an identical copy, right? This new cell has one copy of the red chromosome, one copy of the blue chromosome, exactly what you started with. Start with two chromosomes, end with two chromosomes. And you've doubled the amount of cells you have. You had one, now you have two, and each new cell is identical to the one you started with. So basically, after cytokinesis, each cell is back to G1. After all of this, each cell will now go into its own cell cycle, starting with G1. So this is the end result, okay? Two new cells, each one identical to the one you started with. So this process must happen in order, and it must happen flawlessly. Each step must happen just right in order to get this end result. Things are moving, things are changing. The cell basically completely falls apart. I mean, like, it gets rid of the nucleus, right? I mean, the cell is in chaos during mitosis. Once it gets back into G1, it'll go back to interphase, where it'll start growing, relaxing, carrying out its cell functions, right? If this is a stomach cell or a skin cell that's going through all of this, once it gets back into this format, it's able to start being a skin cell again, okay? It's a very complex process, but it's an important process, and you need to know these steps in order and what happens in each step. All right, thanks, bye.